I'm live. Okay.
You're live? Okay,
yeah,
good.
hold on. But I'm live with a different thing. Fuck. All right.
As long as one of them is working, it's easy. So in half an hour, we can send a link out again.
Yeah. I'm gonna sign you out of Facebook on here, all right?
Go for it. Whoever you need.
Getting hungry. Yeah, actually, that would be great.
Usually see it for the homeless, but today's your lucky day. It's even your favorite flavor, peanut butter and chocolate chip.
Oh man.
I'm gonna send it your way.
Okay. So close.
I just needed a little more behind it.
I know. Sorry. This friggin' snapshot that I took. Hello. Hey.
I'm excited for your talk.
I'm glad one of us is.
Okay. I'll see you in a little bit.
All right.
I'll bring it. What?
I almost ate wrapper.
Oh.
Oh, that's tricky.
There's 3 of you.
So many of me.
Hmm?
So many of me.
Okay. So doesn't the camera have to be like this way?
No, that should be on me.
Oh, you're right. You're right. I don't know why. So kind of. It's a little. Is there a way to
Change the
light? Right, because it's just totally.
washed.
Yeah, like you can't see anything.
I don't know. I don't think so.
I like that. Like, what if we do? What if we pull it back just a tiny bit more and put it on this thing? Or I guess that's not that much better because that's pretty sloped.
Yeah.
Here, why don't
I'm gonna switch desks.
you try pulling it up on your computer or on your thing?
My tablet.
Yeah.
Is it the same link that it was before?
No, you can use that one, but there's a new link on that page for it.
What's happening here? Oh, am I getting your mail or something? I am getting your mail.
My emails?
Yeah. I'm gonna take you off.
Okay.
So I'm so sorry.
Okay.
It's on your Facebook, right?
Yeah.
I'm in two places right now.
Mine's weird.
Like, I'm so glad that he's at daycare because he's probably getting to go down for a nap around now.
Oh, thank God. I hope that's the case.
They said they usually do their nap at 1 o'clock.
Yeah, you're right. We're gonna have to set up the Zoom next to that one, I think, because of Walt. Because he wants to watch it through Zoom too, doesn't he?
I guess so.
I think so.
I would think he doesn't want to log on to 2 different things.
Yeah. Well, he wouldn't have to log on to anything. He would just-
I don't know what the
No.
password is.
Sorry.
Okay, just so frustrating. I don't want to do this right now. trying to see. Okay, so you want to tilt it just a little bit more to the right because it's, yeah, perfect, now it's centered. Yeah, perfect, now it's centered.
I'm just gonna move it back a hair.
Yeah, I think that looks better.
That way people can kind of see me too, hopefully.
See though, the problem is by moving it back a hair, now it's all washed out.
Just wait a minute. It's still washed.
It's better, I can't see you anymore.
Well, I just moved off.
Which were better quality.
Yeah.
That's the only thing that sucks.
Yeah.
Is that it's kind of fuzzy. But, I mean, you're gonna be explaining it the whole time, so. I have to leave your comment.
My comment?
Never mind, doesn't matter.
What was the
Don't
comment?
worry about it.
Okay. Two people are watching. I think it's you and me.
What did you say?
Two people are watching. I think it's you and me. I think I just sent an invite to Wall.
I'm gonna turn this off because I need to put some things. And plus I'm gonna be here. So now you have one viewer.
Just put back this up real quick. Oh, there's 4 watching. People got the hint.
What did you say?
People got the hint. There are 4 people watching. I'm gonna go to the bathroom and clear it back.
I'll guard the cat.
Thank you. Eyes in the sides. Got some water. that thing. That's a good question.
Does it need to have sound?
You know why it's doing it? It's because I have the defense open on my laptop as well.
Oh, yeah, turn that on.
Let's try that again. Let's see if we can get some sound out of this bad boy.
Oh, those damn chickens.
What's up?
I said, oh, those damn chickens.
Hello. Hello.
How are you?
I get water?
You do?
Yes.
Yes, there you are.
Thanks very much.
Yeah, no problem.
I'm Josh. by the way.
Josh, Jason.
Jason?
Nice to meet you.
Are you new grad student?
No, I'm in the staff.
Oh, okay.
So new staff, I started Monday, so yeah.
Like ships passing?
Yes, good luck to you today.
Thanks.
You think everything's up and running?
We'll see.
Brian's gonna be in here soon. He set
Yeah.
up your camera and everything.
Yeah, I think I got the live stream going okay. I'm just waiting on Walt and Janice, who are remotely attending.
Oh,
So,
okay.
Yeah,
Awesome.
so hopefully that will go well.
You've already set up the link, right?
I did. I just forwarded it to you as well to see if it works.
Oh, I have that.
Thank you.
We'll find out.
I think that was you joining.
So it works really well.
Cool.
It's terrifying staff.
I wish I had known it better beforehand, so I could have done a live stream with Zoom, but
I just didn't have time to write together.
no, it's
No.
So we're out there.
yeah. There's a lot of things that would have made this week a little bit easier, I think.
Should I call the parents and find out
Yeah, find out what they're doing.
how they're doing?
They're lost in Ithaca somewhere.
Yes. You're wrong. I'm so. My brother lives in
Hey.
I just did that to
I
myself.
literally just texted you, I was like, I wonder how they.
All right. Not sick at the moment.
He was like, oh,
Very nervous, very shy, kind of shut down a little bit, and then like after.
So how we set up here?
So I've got my live stream going over here, and then I have my laptop here, just for bandits up.
It's nothing.
And then it looks like the wall might have just, there he is.
So cute though,
Just
because
join
it's like this.
Okay, cool.
Hey,
Hey, Brian,
So.
so you need like a whole laptop.
Okay,
That's my direct.
And maybe a DJ table.
Yeah, great. Here, Julie.
Hey, well,
Oh, okay.
So we're gonna be able to project.
let's try that.
Oh, great, thank
Mike
you.
is just bigger. I
Yeah,
started
and
on
Mike.
Monday.
Yeah, nice to meet you.
Meet you, so,
Welcome to the team.
thank you very much,
Okay.
thank you.
Okay, and then that will,
Yeah, let's turn this off.
and I have the directions for how, where the, should I kind of take it apart? Should I carry it like that?
Yeah,
Secure?
Yeah.
With this secure here, so that's,
Yeah.
I don't want to break anything.
Oh, yeah, go right
Hold
ahead.
this up. Oh.
You know, this cable is pretty wonky. Hey, Brian.
Yeah.
You know, we've been having tons of trouble with this cable, right? This HDMI.
Yeah. Are you using HDMI?
No. Yeah,
Oh, you're using HDMI.
Yeah,
I
that,
don't know if this one has HDMI.
yeah, I mean, yeah, so that should work fine.
Okay.
But I'm getting new cables to switch this out. Yeah, so what I think is the problem is, these cables are a little bit, well, this one's not even bad, but, I feel like the weight sometimes on certain laptops, yeah, you know, and it's really hard, so I got an ultra thin cable, I'm gonna try that, but the other thing I got, and just gotta let everybody know is that I think an adapter, an HDMI adapter, like for USB or something, and then put a cable in. So I think it happens like when it's directly plugged, mostly I think, but
All right.
it's just like,
Well, I think it's working now, so
Yeah,
why don't you
yeah.
see if you can put walls up there? Gonna move your wall?
We can try.
Oh, yeah.
Oh, I guess, probably just do mirror rather than
Okay.
whatever it is.
Switch this mirror and then I come on, is that right?
Yeah, we can see you.
Okay.
Yeah, we can see you just fine.
Yeah.
Now let's see what happens when can we put this on?
Yeah.
Center up. Yeah.
I just moved it here.
oh, okay.
So not to move that, it's too much stuff.
Yeah, just transfer over the one file that's on here on your desktop. And open it up.
You Dougie, I think I'm gonna move you. Yeah. But you wanna hear the seminar? Huh? You do? Huh. I had no idea Diesel was interested.
Okay, then you can reject that.
Do we have to watch Walt?
I'm afraid
That's just the defense, it's Walter Diesel. Lost. Sound. Oh, there it goes. Yeah, it popped out for a second.
Hey, keep going. Bye. Bye-bye. Ah. You can move it next year.
I don't know if I've actually ever used PowerPoint this one before. Oh, man. You went to YouTube or Facebook, didn't you?
That's smart. Now I wonder.
So what I'll do is I'll do the intro, I'll show my thing, okay. Then I'll close this.
Sure, just
And then we'll repeat it. Okay. So you just have part of the intro, sounds good. And then after we're done, you switch to your, right? Yeah, okay. So maybe we should.
No, I think Walt took over the screen.
What'd you do, Walt?
what did I do?
I don't know, just took over the screen though.
What did that do? Did that do something?
Yeah, now we can only see you.
Yeah, it shared desktop, but then I did it again. That's all.
Yeah, you don't want to share your desktop. Try turning that off.
Okay. Stop share.
Okay.
Okay, now it's actually should be shared.
Should I try switching back to the side to side?
Yeah.
Let me see if that helps.
Tony Tracy.
Okay, we're working on it here too.
Tough just. But she's gonna do that.
What are you doing at the moment?
Hang on, we're messing around with things.
Yeah, I won't touch anything. I'll let you guys do it.
That.
Okay, he's up there.
Okay.
Now go to PowerPoint.
Which is what they have.
Now let's see what happens if I.
Alright, it's got,
I'm gonna come out over
it shows
here.
up in your.
Okay, cool.
Josh.
So then,
We're gonna be looking at that for them to see.
I am.
We're not there yet. It's for your two.
Do you really want to broadcast what Walt can discuss?
I'm turning him down.
To be sure some of this they would.
Here, back.
My daughter is like 10 miles from Princeton.
Where?
I'll see you in January.
Cool. These are my parents.
Say hi.
Hi, nice to meet you. Kevin. Oh, okay. Yes, it's a yes.
Oh, that man. Okay, great.
Yeah, exactly.
Oh, really?
Just go on, take time.
Yes.
I will get in on Saturday.
Fun seeing you.
Yeah.
It's here.
Yes, it is. Yes.
Yeah. So. We're good. And then Walt, can you still hear us?
Thank you. Thank you.
Oh, right, yeah, do we hear it?
Yeah, we can hear you, but I'm gonna turn you down for a minute.
Okay, can you hear us?
Yeah, we can hear you, but actually, why don't you?
Screen sharing this thing, so we'll see your talk when it comes to that.
No, you'll see it, how will you see it?
I guess we'll just have to turn it a little bit because the camera's still working, right?
Ooh, I wouldn't have done that.
But I just lose. Just a little jiggle. That's all it takes.
Still not gonna work.
Okay.
Okay.
You got something? By the way, you have a black screen, right?
Yeah, but that's.
Do I stand up to the side?
Yeah, or stand up the side, whatever.
One gets lost.
It's not Sabrina, I don't know what it is.
That's what happened. Yeah, I mean. I'm just a doctor, really good at that.
It's a great question. Question for the ages.
Yeah, that's a really one of those.
Yeah. Usually it's back probably.
When you go, are you going back to the college?
Yeah. I did, dude.
I did.
I think the only
There.
I'm going back for a class.
Too long. Okay, I'll look at it.
We did on the beach. Very flat. So you can smell that one. You don't smell it. They have some extreme they're really getting hit with the whole rise.
It's a great divide.
How about you?
Yeah. Oh, really? Yeah, it was yeah, and that's everything else. All right, welcome everybody. I'm glad to see so many were able to make it for this unusual seminar hour, where we're here to hear about what Josh LaPergola has been up to for the last several years since he first came here to Cornell in 1929. So when Josh first came to Cornell, he started as a student with Paul Sherman and Walt Koenig. More recently, he's moved into my lab with the retirement of both of his other advisors. I suddenly feel like retiring as well.
Just one more thing.
And when he arrived here, he took on what many in the room know is extremely challenging study species to try and untangle and understand the various mechanisms and selective factors that shape coloniality in the species of woodpecker. And he did face all sorts of challenges in doing this. He faced those challenges with ingenuity, integrity, tenacity, I think was a word I would use, and basically conquered them all to come back with an armload of data so we can help understand this really unusual and interesting bird and why it nests in such an unusual way. As I said, Josh joined my lab fairly recently, but it's been really nice getting to know him better since he's joined my lab. And as I've said in many of these introductions, what's really nice about getting to know a PhD student is you learn a bit about their science and you help guide them do that, but you also learn about other aspects of their personality. And in Josh's case, I learned that he has a very artistic side to himself, in particular with respect to the musical arts. In fact, I learned that he is a dancing machine as well as a good scientist. And you can see this displayed here. Apparently it was displayed from a very young age. I glanced at this picture and I didn't even have to look closely at the facial characters or anything. I knew instantly which one of these was Josh. But then he grew up and really perfected some of his moves, better and better. And then when the sort of single ballet type maneuvering wasn't quite enough for him, he joined an all-male Nutcracker Suite ballet band that performs every holiday season, I think, right? Sure. State Theater? Okay, good. Looking forward to that. And it's not just the physical dance parts of music that Josh has performed so well at. He's also mastered other aspects of the musical arts, which I think our co-introducer will be telling us about if we can get him on the screen. Look at this one. That's the wrong one.
It's a little bit.
Yeah, let's try it this way. Do you have any idea?
Try that.
Oh, I don't know.
Maybe not.
Yeah, okay, let me try this.
Oh, that sounds good.
Hello?
We hear you.
Do you? Ah, well. Okay.
Oh, I think, are you sharing your screen, Walt?
Let me try that. Does that do something?
Oh, yeah. Okay, perfect.
Really? You got me?
Yeah.
Hello, everyone. Greetings from Central Coastal California. I do want to thank, first off, Mike for taking over for those of us who decided that it was time to get back to our roots and ride our bicycles instead of shoveling snow every day. So yeah, I want to just say a few other words about Josh. Josh came in, as Mike said, it wasn't 1929, I think it was 1829. We enjoyed every minute of it. He had come from Rowan University. You guys actually get my screen? Oh, there, yeah, you do have it.
Yeah.
I'd never heard of Rowan University, but it turns out they're the Fighting Owls, or the Very Upset Owls. And so it presumably got Josh interested in birds at an early age. As I think Mike mentioned, he then went on to Villanova and studied black catbirds. The fact that he had experience in the tropics was part of what prompted us to think that he might be capable of doing a project that I always thought would be extremely interesting. Working on Hispaniolan woodpeckers. He'll tell you about why they're so interesting. But it is a bird that there's been a lot of work on in the past, but none of it has been very interesting. So I figured it was time to do interesting work on it. Okay, so yeah, there he is in his musical early days. What Mike didn't mention is that he was actually in more than one band. This is one that was called, I don't know, what was it? It was something like Dirty Josh or something like that. And this is him playing the guitar, but what I really wanted to show you was that there is actually a YouTube video of this. See whether I can get that up. There it is. The best part about this is that this is what apparently passed for music in southern New Jersey. Let me see, I think I can get this. We're probably going to get something weird here. I won't bore you with it. The best part is that there's this kind of weird stuff coming from the band for a couple minutes. And then at the end, Josh starts jumping up and down for no obvious reason. It's really kind of the music. So if you want to actually see this, I recommend all of you go back and Google Josh LaPergola weird band. It's really kind of amusing. Okay, let's see. Yeah, so then there were some early signs that Josh would be interested in studying a bird that lives up in trees. He got used to wearing a helmet, which was a good idea, as it turns out. And the other thing he started trying to learn how to do was to fly. And I wish he'd gotten better at that because it might have saved a lot of us a lot of trouble, including him. In any case, We realized that if he was going to study birds, he was going to need a lot of help. I did come out and visit him back in 2014 in the Dominican Republic. He really had a great setup there, and we're all looking forward to telling us about it. So, without further ado, allow me to introduce Josh LaPergola. Okay, go for it.
And before you get started,
Combination of many years of dissertation research, the ambient mode.
this is the reason we do it all. That was awesome. Thank you guys for that very disarming introduction. For the record, yeah, Rowan is the professorial owls, which is much more intimidating, I like to think, than a fighting owl. And my band was a ska band. We were called Dirty Larry. Thank you so much, everyone, for being here today on this cold November day. Some of you are far away in warmer climates, but hopefully the rest of you are feeling nice and warm and can be whisked away with me to the tropics to learn about what I will contend is the most fascinating bird in the world. And Walt isn't here to contradict me on that, so we're good. But the main thrust of my dissertation has been trying to understand the forces, the causes and consequences of colonial nesting, a really interesting form of social nesting behavior that I'll tell you a little bit more about. And so the structure of my talk today will follow this form. I'll give you some background about colonial nesting itself and my favorite bird in the world, the woodpecker. And I'll be using these little photos to kind of guide you throughout rather than giving you the formal intro methods results, chapter 1, chapter 2, chapter 3. But the next 3 bullets roughly correspond to the chapters of my dissertation. The next question I try to answer is what drives the variation in colony size in Hispaniola and woodpeckers, whether or not Hispaniola and woodpecker colonial nesting is adaptive or possibly forced due to limited resources, and how this bizarre social nesting system actually impacts their mating strategies. My interests mainly are driven by trying to understand why variation in social behavior exists, in particular group living. At one end of the spectrum, we have organisms that live mostly solitary existence. It's like this jaguar here. At the far end of the other side are eusocial organisms which live in complex societies and have castes and division of labor. But what really drives me is the stuff that lives in between, the stuff that still maintains its autonomy, but yet aggregates in time and space. And these are the colonially nesting species in the world. And they drastically vary in the amount of diversity they show in their social behavior. For example, these penguins up here are just aggregating and not really doing much, maybe cuddling for some warmth. And these white-fronted bee eaters here, made famous by Steve Emlen and crew, have very complex social relationships within their colonies, including family groups. So colonial nesting itself can take a lot of forms in the bird world. We can have very simple scrape nests like these terns up here where you just have a bunch of birds hanging out on the beach and laying their eggs there. We can have some frigate birds here that are aggregated in a clump of what I think is mangrove. But then you can also have birds that are putting their nests in places that seem like there's no really good place to put them. Things like pendulas, sociable weavers, and palmchats, where they're aggregating their nests on a tree that seems like it's no better than any other tree, perhaps. And one of the key and fascinating things, and things that plagued biologists for a while, is that there's tremendous variation in the size of colonies across species. This is best exemplified perhaps by the cliff swallow, which has been a workhorse for the study of colonial nesting, where you can have singular nests and colonies up to thousands of nests just punched together on the side of a culvert like this. And so these species that have singular nests and massive colonies all together in one population are fantastic because they allow us to make use of natural variation in aggregating behaviors in these birds to test the hypotheses for why they do these things. Which brings me to my favorite bird, the Hispaniola woodpecker. So there are a couple of great reasons to study this bird. One of them is not that they're easy to study, certainly, but they're one of 3 colonially nesting woodpeckers out of a family of over 250 species. So roughly 13% of all species of birds are thought to be colonial nesters, but this comes out to less than 2% of all woodpeckers. So very underrepresented social behavior in a family that's fairly large. Begs the question, why this species and the other 2? They're also facultatively colonial. So within a single population, you can find both singular nests and colonies that can range in size in my study site up to 12 pairs in a single tree. There are some records from the 70s and 60s suggesting that colonies can even get up to as large as 26 pairs in a tree. Not as large as a 1,000 nest colony of cliff swallows, but still certainly a lot of natural variation that we can play with to try to test some hypotheses. So I'll whisk you away now to Hispaniola, the home of my friend the woodpecker. I do all of my work here in the Dominican Republic, which takes up roughly 2/3 of the island of Hispaniola. The woodpeckers found in both Haiti and the DR. We zoom in a bit. The woodpeckers found pretty commonly up to about 2,000 meters above sea level, but I did most of my work here at a medium elevation, about 500 to 700 meters above sea level, where they're fairly abundant and have a fairly long nesting season. As we zoom in a little bit closer, most of the work that we did was focused on private land, which was a patchwork of the savanna, palm savannas and grazing pastures, and then secondary forest fragments, consisting of various broadleaf and also pine trees. On the ground, it looks a lot like this. We have a bunch of these royal palms dotting the landscape, forest fragments, and the woodpeckers love royal palms. In fact, if you were to go back to some of the earlier literature from the turn of the 20th century, you would find people were commenting on how much the birds appeared to love palm trees. At lower elevations, you'll find them often in cotton palms, which are another palm species, and also coconut palms, especially along the coast. But at this mid-elevation site, they apparently are in love with royal palms. Now, because they're a woodpecker and they're nesting in palm trees which have no branches, that means we have a special challenge. If we want to understand the impacts of colony size and variation, what's driving that variation, we need to get into those nests and see what's happening. Unfortunately, most of the nests are about 10 meters or higher. So just for some scale, that's me at the top of that palm tree. It's roughly 60 or 70 feet up. Now, on a good day, it'll look something like this. You can get to the top of a tree or to all the cavities on the way up, and we can directly inspect inside the cavity to get the nest contents, number of eggs, etc. On harder days, or maybe more tiring days, we'll use what's been lovingly called a pecker peeper, which is a wireless camera that we can insert into cavities up to about 30 or 40 feet. And we get these grainy images that we can use to determine things like hatching success, clutch size, etc. The next step we take in our methodology to study these birds is we have to mark them. It's not really easy to determine colony size if we don't know how many individual birds are in a colony. These woodpeckers are nice because, like many species of woodpecker, they're sexually dimorphic, so we can at least tell males and females apart. But when you get to have like 20 or 30 or maybe even as many 40 birds on a single tree, there's a lot of commotion going on, so it helps to actually be able to identify them. So we took a technique used for acorn woodpeckers, which involved cutting into the side of the tree to make this little trap door, and then ambushing the birds when they went in to sleep at night or when they went in to feed their young. That didn't work great. It works great for acorn woodpeckers, I think, and if you are willing to have a small sample size, it can work well for Hispaniola woodpeckers. But I needed to kind of ramp up the situation. As Mike alluded to, this was the tenacity that came into play. We had to make these ridiculous mist net towers to actually catch appreciable numbers of birds. And so here you can see there's one woodpecker here, but I'm telling you, it did a really good job. And just as proof, just last year, these aren't woodpeckers, unfortunately, these are palmchats. But you can see there are actually 19 birds in this net that was up for less than 10 minutes. So it's great for woodpeckers, phenomenal for palmchats. And if you'd like to use it, come let me know, because I got plans. But that's all to say, we need to catch a lot of birds and mark them so we can know who everybody is. So everybody gets these dope bracelets so that we can check them out and individually identify them. They come into their nests. Usually we can get a pretty good look at least one leg, if not both. And if the angle's right, if they come back repeatedly, we can figure out who's going where. So that's the basic gist of what I did to try to answer the questions I'm about to tell you. So the first question that I think is key to understanding coloniality, and one that again has been troubling behavioral ecologists for a long time, is understanding what actually drives colony size variation. So again, there's a lot of variation in colony size, and people have tried very hard to explain it, and it's been really difficult. In particular, a lot of species will nest in one place and not in another, and they seem to human observers to be no different. There's one really simple and elegant possible explanation for coloniality or colony size variation, which is this habitat-mediated aggregation hypothesis. This is roughly equivalent to the ideal free distribution. So the idea is that the distribution of resources on the landscape is roughly a really good predictor of where birds are settling in order to nest. And so because resources are really packagely distributed, you get nests that are clustered as well. This was really well exemplified by redneck grebes, which nest on these floating islands. And this great study where they found that as time went on, the islands started to tear apart, and the number of nearest neighbors that a particular nest had also decreased over time, concurrently with the decrease in the size of these mats. But there aren't many very good examples in the bird literature of species where this applies. But it's possible that it could work for Hispaniolan woodpeckers. Now, let me tell you why. We know already that they love royal palms. So one possibility is that royal palms themselves are limiting on the landscape for these birds. If that were true, we would expect to find one or more nests in all palms, right? The second possibility is that palm trees of a particular status or condition might be really good and preferable over trees of another status or condition. In Hispaniolan woodpeckers, it's been suggested that they really like dead trees, possibly because they're really easy to excavate. So if this were true, we would expect colony sizes to be much larger on dead palms. We would expect that colony size should increase once a palm died, and that cavities in dead palms should be superior for one reason or another, and I'll tell you what that reason is, and that they should be easier to construct. It's also possible that cavities can accumulate over time, and that could be something that can help drive colony size variation. So a particular cavity, for example, could be constructed in 1 year and then persist for maybe as long as 10 years. We don't know yet the dynamics of cavity duration in this particular species, but in some regions of the world, for example, I believe in British Columbia, you can have cavities that can exist for over a decade, and birds can still use them. So these three different main drivers, royal palms themselves, dead palms, and the aggregation or clustering of cavities, might help explain colony size variation in this species. So the first thing I did with my wonderful teams over the years is we tried to mark as many palms as we could. In 1 year in particular, we did an exhaustive survey of a relatively small patch just to find all of the palms in it and figure out whether they had cavities or not. So not necessarily current use by woodpeckers, but at least past use or possibility of current use. In addition, because the landscape is fairly large, I turned to Google satellite imagery to try to quantify the number of live and dead palms, because there might be a really small number of dead palms in one particular patch, but when you go out a little bit further, it might not hold true. And so these aren't particularly the greatest examples, but they are examples of what you might see in Google satellite imagery from 2013. This is a live royal palm. You can see it's got this kind of star-like shadow. And then more recent imagery, you can see it's got a much more characteristic shape. And then there's a dead palm nearby, which has a long shadow on the landscape, and you can start to make out the details of the dead palm there. Here's a slightly more clear example, and this is one of the beautiful things about Google satellite imagery or historical satellite imagery. You can actually see which palms have died. Go back in time to figure out if what you think was a palm actually was. So this particular palm, this is from 2017. You can see there's a little bit of a snag there. If we go back to 2009, we know that it was a palm because it looks like other palms that we know were real palms based on ground truthing. So based on the exhaustive survey, we found that only 14 of 37 palms actually had any cavities. So pretty small sample size, but pretty conclusively says that palms themselves aren't going to be the limiting factor and something that could drive colony size variation. And in particular, in that patch, there was only one live tree, or sorry, one dead tree. The rest were all live. But based on the Google satellite imagery analysis, we found that roughly 94% of the royal palms that we could identify were live. So that would suggest that dead palms are much rarer on the landscape. Anecdotally, I've marked 22 dead palms in my time in the last 6 years, and every single one of them had one or more cavities. Most of them seem to be used at the time. It's possible that, for reasons I'll get into later, the longer the tree's been dead, the less useful it might be. This is a great side-by-side comparison of live and dead trees. So remember, it might not be palms themselves that are limiting, but trees of a particular condition. That's exactly what we found when we went out and actually measured colony sizes, the number of pairs using different trees. So this is my favorite set of trees. This is Hansel and Gretel. Hansel is the dead tree, Gretel is the live tree. When we first found these trees in 2012, they were alive, so they were a nice additional experiment there. If you look at Gretel, in this image from 2013, there was only one pair using that tree, whereas on Hansel, there were actually 11 pairs, all nesting on the tree at the same time. In addition, something that seems to make nesting in dead palms superior to nesting in live palms is that roughly 30%, 29% of all nesting attempts on live palms failed because of cavity flooding. So either partial, where the nest gets a little bit soaked, or complete flooding, where the eggs and nestlings are completely drowned. Whereas on dead trees, on dead palms, we never saw water accumulate in a cavity and cause nest failure. It's intriguing, right? In addition to that, cavity excavation on dead palms, for a small number of nests we were actually able to measure this, only took about a month on average, whereas on live palms, on average, it took about 2 months, and sometimes it could take over a year, possibly because birds were starting and stopping and other things were going on. Another possible benefit to nesting on live palms, if you think about cavity construction taking less time, that might be good because they could start nesting earlier. That's not the case. So there really isn't a difference in terms of the average time of initiation of the first nest in a year between dead and live trees. But new cavities, which are mostly built on these dead palms, do start later than those in old cavities, which seems like there was a tendency for more old cavities to be used on live palms. This wasn't significant in any of the models that I used to try to tease this apart, but it could just be because the interaction that we would expect to see is not detectable statistically. These little green dots correspond to the newly constructed cavities in live trees, and so I don't really have a large sample size, mostly because what would often happen is, and most of these nests didn't get into the analysis, the birds would construct a cavity, or try to, in a live tree, and then it would flood. And then they would abandon the attempt or move on to greener pastures and nest somewhere else in the following year. So when we look at the colony size across the study population, what we find is that dead palms do host larger cavities. So roughly 5 pairs is the average colony size, but it could be as much as 12 pairs in my study set. Live palms, on the other hand, can only as far as we've seen, only hold up to 4 pairs on a single tree. This relationship is in part driven by a difference in the number of cavities that exist on a tree. So here we have the number of complete cavities on a tree and colony size. You can see the relationship is much closer to a 1-to-1 relationship for live palms than it is for dead palms. And part of that is because a lot of the cavities that would seemingly be fine for a nest on a live palm are actually not available to the woodpeckers. And that's because some of them, about 20 and 30%, are occupied by heterospecific, possibly competitor species, especially kestrels and these Jamaican fruit-eating bats. But a lot of the cavities on live trees are flooded for a part or all of the year. And so you can have possibly commensalistic organisms like tree frogs making use of them, and then also some aquatic larvae. But the take home is that while there are a lot of cavities on live palms, most of them are not actually available for the woodpeckers. Something that I wasn't able to really tease apart is that I think an interannual variation in precipitation could be driving some of the colony size variation, because in dry years, some of these cavities will actually dry up and be used for nests, whereas in wet years, you'll never see a nest in one of these cavities.
Josh, who makes the cavity?
These woodpeckers do. Sorry, that's another really important point. Thank you, David. There are only 2 woodpecker species on Hispaniola that are resident. There's this one, which is a large Melanopes woodpecker, and there's a small piculate, which is not very social and not nearly as common. So these are the predominant producer of cavities on the island. When we look at the breakdown of the availability and usage of these cavities, you can see that on dead trees, over 80% of cavities that we monitored over the years were actually used for nests. Whereas on the live trees, it's only about 30%. So tons of cavities out there, but not all of them are being used. And again, that's because over 40% of these live cavities, or cavities in live trees, are flooded, sometimes for multiple years, in fact. And the relative unuse category, these were cavities that weren't possibly monitored quite as well, and there could have been other reasons that made them less attractive to woodpeckers that we didn't have time to address, or maybe the resolution to really understand it. But this all suggests so far that dead trees, dead palms are really attractive to the woodpeckers. And so the next thing that I did was I looked at colony size change between years as a function of the condition or status of the tree. And so this is a slightly complicated figure, and I'll walk you through it. All these bits in green are the raw data for live trees, and the model predictions are these lines. So you can see for live trees, colony size has a slight tendency to decrease from one year to the next. And that's kind of irrespective of the number of cavities that were present on the tree in the previous year. Whereas for dead trees, there's a negative relationship between the number of cavities on a tree in the previous year and colony size change. So what that means is if there are a ton of cavities on a tree in one year, the next year, colony size is more likely to go down. And that's because on these dead trees, as more and more birds nest on it, they can actually erode the inside of the tree, such that after a couple of years, you can only have one platform that can sustain a nest. But trees that just died, when they had no cavities, they were colonized the next year. And those that died that already had cavities had the largest increases in colony size. And both of these pieces of information suggest that it's not just that dead trees are great, but it's also that these cavities that are already out there can be used once they become drained and available. So all of these pieces of information together are suggestive of the fact that colony size variation in the Hispaniola woodpeckers are driven by the relative rarity of dead trees and the availability of cavities on the landscape. And this seems to be possibly pretty unique for colonially nesting species in general, and possibly for woodpeckers. It's really hard to find good data on nest failure due to cavity flooding. Possibly because it's underreported, or possibly because it really is just rare in most species. Now that we have kind of an understanding of the factors that are helping to drive variation in colony size, it would be really good to know whether being in a group itself is actually beneficial. And so biologists have known for a long time that group living carries a lot of automatic costs. This goes back to Richard Alexander's landmark paper. You get a lot of parasites and exposure to pathogens by being in a group. There's more competition. You can lose out on mating opportunities. But if that were always the case, that there were a detriment to being in a group, then you would never see it. So what we understand now, of course, is that there have to be some benefits to offset those costs. And in some cases, they're more than enough to favor coloniality. Some of these social benefits might be things like predator, or reduced predation effects. So if you look at this picture of all these Caspian terns, probably if you've ever been on a beach where there are terns nesting, you know that a large swarm of birds flying around, pooping on you and dive bombing you is a really good deterrent for predation, even if you're not really a predator, if you're just a hapless beachgoer. So that's one possibility, is that you can get defense against predators for you or your young. Another commonly cited social benefit is social foraging. So you could potentially follow knowledgeable individuals to good food resources, or maybe by working together, you can get better access to good food. So this makes me wonder whether colonial nesting could possibly be adaptive, be driven by social benefits in the Hispaniola woodpecker. Like I said before, there are only 3 species of woodpeckers that nest colonially. Only 2 of them do it very frequently, this species and the Andean flicker. The other, Lewis's woodpecker, does it occasionally, and not to the extent that this species does. But there's quite a lot of conflict that appears to go on at these nests and at these colonies. So this tree in particular was really interesting. It's a live tree, and we found it just at the time of colonization. So this was in 2016. Birds were just beginning to settle on this tree. They eventually constructed 1 cavity, and 1 pair took it over. But that doesn't really explain why there are a bunch of birds hanging out of it. I think that birds are fighting over good nesting spots. And I can tell you more about that at the end, but that's a little bit further afield from my dissertation focus. But all that is to say is that it's very common to see birds fighting at colonies, and also it's very possible that it's not just a harmonious and lovely coexistence for the species in these colonies. So the alternative possibility to social benefits selecting for coloniality is that birds are forced to live in these aggregations. And we can distinguish between these 2 possibilities based on the patterns of reproductive success that we see associated with colony size. So for my work, I focused on all these correlates of reproductive success at the level of the nesting attempt. And then for a subset of pairs, which I was able to follow very intensively within years, I got measures of annual nesting success and annual fledging success. And this is one of those moments in your dissertation when you're presenting and you just want to cry, because there's so much statistical magic behind this, like large tables that I'm not going to show you with. And that's because if we look at these patterns, fledge size isn't statistically related to colony size, neither is hatching success. Probability of losing 1 or more young before fledging, the number of young that you fledge. Annual nesting success is also not related to group size at all. And it doesn't matter how you parse the data, if you're looking for a relationship between colony size and these things, or singular and colonial and parsing it that way. None of these are statistically significant, like way not significant. That's a very scientific term. The only thing that did show an intriguing relationship was nesting success, and this is the binary response of whether you fledge any young or whether you fail completely. And I say this is interesting because the the association between colony size and nesting success is positive. But when you look at the raw data and what the models are predicting, I don't totally believe it. And I would encourage you all to be skeptical as well. Not because I'm bad at science, but because these birds are weird. So the colonies in dead trees are marked in blue. The colonies in live trees are marked in green. And if you'll remember before, I told you that live trees only ever hosted colonies of 0 to 4 individuals. 0 to 4 pairs. These two bizarre bars here are actually that couple of trees I showed you before, Hansel and Gretel. So it's possible that they are messing up the interaction term between tree status, tree condition, and colony size. And if you actually pull them apart and reparse them out, you can see that there's a negative trend kind of showing up here on live trees and kind of flat for dead trees. I think that this relationship is more driven by the fact that there is variation in how long the trees have been dead, and variation in the causes of nest failure between live and dead trees. To give you a sense for what that looks like, this is also a busy graph, so I'll walk you through it. We've got singular nests in dead trees, singular nests in live trees, and then colonial nests in dead trees and colonial nests in live trees. And remember before when I told you that flooding is a major cause of nest failure? Well, that's still the case. It's really important for nests in live trees. And in fact, almost half of all failures in live trees are due to flooding. Very little is due to what we would call predation. And I should say that there's an asterisk after predation. We never really see predation. The terms that the observations that I have lumped into predation here are those where the eggs disappeared inexplicably, nestlings disappeared. They were just gone in between checks, and chicks definitely weren't old enough to fledge. So it's possible it could be a snake or a rat or something like that. But intriguingly, the few cases of nest destruction we actually saw were by other Hispaniolan woodpeckers. So it's possible that one of the things that balances out being in a good dead tree is actually birds fighting over the cavities and potentially killing each other and killing each other's young. So if we look here at the predation, we can see that the proportion of failures, the percentage of failures in colonial dead trees, that's most of the failures coming from this predation category, whereas in colonial live trees, it's much lower, and there's a lot of flooding in contrast. Same thing when we look at flooding, a lot of flooding in singular live trees and colonial live trees, none in colonial dead trees, and in this weird singular dead tree case of flooding, it was actually a non-palm and the nest just got soaked. So it didn't actually collect any water, it wasn't a palm tree, so what I said before still stands. But this is all to suggest that there probably aren't very many benefits to being in a colony for these species, and in fact, the results that you more or less have flat or no relationship between colony size and reproductive success, that's more consistent with resource limitation. It's not definitive, but it is very suggestive that what's actually going on is that cavity flooding and the rarity of dead trees in the landscape is not only driving colony size variation, but it's actually making it a better choice to just nest near other birds than to not nest at all. There's one more subtle way that coloniality can impact the biology of a species, and that is through the mating system. And the way that I've approached this question when it comes to the Hispaniolan woodpecker is by pitting two alternative hypotheses against one another. And it'll make more sense momentarily. But broadly, I'm calling these the opportunity hypothesis and the parental care hypothesis. And my beard tip friend here is my example for the opportunity hypothesis, which is the idea that as the local breeding density increases with increasing colony size, there should be more opportunities for birds to get matings outside of their pair line. So the Hispaniola woodpecker is a species that is in pairs. They don't have a very complex mating system. They don't have cooperative polyandry or polygynandry like acorn woodpeckers do. So it's possible that by being in a large group, they might have more opportunities to lose and gain parentage. So this can come through either brood parasitism, where other birds in the colony or outside the colony are dumping eggs into their nests. It could be quasi-parasitism, where a male allows an extra pair female to lay an egg in his nest because they've mated. Or it could be through paternity loss, an extra pair paternity, where a female mates with other males in the colony or outside the colony. Contrary to that, the parental care hypothesis postulates that the high need for parental care can actually select against paternity loss or parentage loss in general. This mostly stems, the way I envision it, comes from a game theory model developed by Reed, and is potentially applicable to both brood parasitism and paternity. The idea here is that in species where there's a high level of investment by one or both sexes, that sex should be under intense selection to prevent any kind of parentage loss. And people have argued that in woodpeckers, males actually invest quite heavily in their nest and in their young. So as far as we know, in all woodpeckers where it's been studied, the male shares roughly half of the incubation throughout the day, but then he also incubates the eggs and the young overnight, whereas the female is doing something else. Karen Reeb has actually suggested that in northern flickers, they have reversed sex roles, and that females have been somewhat liberated from their parental duties, and that's why in that species there's actually a high incidence of conspecific brood parasitism, where females are dumping eggs in other nests. So makes us wonder what's going on in Hispaniola woodpeckers, because they're colonial, and so they should have opportunities to seek extra pair copulations, but they should also have opportunities to dump eggs, even if the males might not want them. So to do this, when I was catching adults and while I was sneaking around in nests, I took blood samples from as many young and as many adults as I could. Through the wizardry of modern science, I brought these samples back to the US, extracted them and parsed them down into smaller and smaller numbers of vials, did some wizardry in bioinformatics, all thanks to the Fuller Lab and Robin Butcher, many thanks to you. And this is another one of those examples where a lot of hard work can just be summed up in one slide. What I did with all of these data, all this effort, is to develop or identify single nucleotide polymorphisms, a type of genetic marker that you can use to do parentage analyses. And so when I do those parentage analyses for 71 young from singular nests, 175 young colonial nests of various sizes, it turns out that every single one of those young can be assigned to their parents with high confidence. So it looks very much like there is a crucial role for parental care and its importance in the Hispaniola woodpecker. And just by having lots of potential mates around you or potential parasites around you, they don't really suffer very much. It is also important to note, though, that my sampling was kind of biased towards successful nests, and we tried to get some unhatched eggs, but there's quite a lot of hatch failure in this population. Most nests have one or more eggs that don't hatch. And so it leads me to wonder whether there could be brood parasitism happening in this population, in this species, and we're just missing it completely because we didn't sample enough young. I would say, though, that based on the genetic analyses of young, most of which made it to fledging, we can say pretty confidently that they never fledged parasitic young or extra pair young during the course of the study. So it would seem that either they're just really bad brood parasites, or that, and by that I mean we never detect it, or that they have really intense selection pressure to not go outside their pair bonds. So, my friend, the Hispaniola woodpecker, what's going on there? I should come back to this point Walt made earlier, which is that he said a lot of people had done some work on them that wasn't interesting in the past. I would say it was interesting, but one key thing that they were missing was they weren't focusing on coloniality at all, and they were doing all their work on unmarked birds. So I think now that we're starting to have a better idea of what's driving coloniality in this species, I think hopefully more people will be encouraged to go out and study coloniality and colonial nesting in lineages where it's extremely rare. So a lot of work has been done in the swallow family and in shorebirds where it's very common, but in families like the woodpecker family where it's rare, it's really understudied. And so we don't know how general the explanations we've derived from these other groups are. But at least for Hispaniola woodpeckers, it seems like resource limitation and the distribution of resources might be really key for driving colony size variation. So far, it doesn't seem like there are any benefits to being in a colony, though it's possible that there could be impacts on adult survival, and that's something I want to look at in the future. But I think one of the coolest parts is that there's no impact on their mating system. So the mating system of woodpeckers in general haven't been that well studied, but the few that have been, most of them don't have any extra pair paternity, so they're fairly faithful to the mate. And with the exception of the northern flicker, there's rarely brood parasitism. It does happen occasionally, but it's not as well documented. And so only, I think, about 10 species of woodpeckers out of the 250 plus have been subjected to mating system study, and I think hopefully once I get this published, more people will think that it'd be worthwhile to look at some of the other species, especially the Andean flicker. Somebody needs to study that bird. That's my dissertation in a nutshell. And with that, I have to acknowledge my funding agencies and all the people who really took a bet on me doing this ridiculous study, because these birds are hard to study. In particular, my thesis committee has been awesome and super supportive. Herbie Levitt, who was originally on my committee, also couldn't be here today, but he contributed a lot along the way, always pushed me to be really thoughtful about the approaches I was taking. And Paul Sherman early on as well was really important for the development of this project. Bronwyn Butcher, as I mentioned, was just super patient with me in the lab, and I have to acknowledge her for all her help. And then a lot of people really helped with the logistics of getting this running. And it would be remiss of me to not acknowledge the incredible field crews that I had over the last 6 years. So starting back in 2012, where I only had one person, all the way up until 2017, where I actually had 9 people, which was just mayhem, but awesome. They were really crazy people who love birds and love being in the tropics, and I couldn't have done it without them. They're amazing, and I know a lot of them are pursuing science, and they put up with a lot for me on my best and worst days, and especially the 2016 crew. But I'd also like to take a moment to acknowledge my committee a little bit more directly. Janice Dickinson, who has been with me from the beginning. First of all, I love this photo. I just Googled her online and found it, and I don't know why more people aren't using this still. But anyway, Janice has been great because she's always forced me to be very clear in articulating my hypotheses and thinking deeply about the assumptions and the approaches I was taking with this work. Curran has also been instrumental in making me sure I make my assumptions very clear and making me think about things from a modeler's perspective, which has definitely improved my scientific process. As was mentioned previously, Paul, who retired right before my A exam, is no longer on my committee, but he was so important to me early on when I was forming this dissertation and the project, being a tireless cheerleader and always asking me, am by falling in love with my study organism. And I think I can say pretty confidently that I have fallen in love with Hispaniola woodpeckers, although I'm not sure when I'm going to get to see them next. Hopefully soon, though. But Mike was very gracious and stepped in after 2016, which I'll talk about in a minute, and really helped me bring everything home. And Mike, thank you so much for your patience and your diligence and just everything you've done. I think I might have told you this already, but I've definitely remarked it to other people that I probably should have had you on my committee from the get-go, because there's so much overlap between what you did for your dissertation and the ideas and things that inspired me to do mine. So I'm glad that it worked out in the end that you could be a part of that. Thank you. And then lastly, Walt, who I'm not sure if he's still with me. Hopefully he is. Actually did come down to the field with me for a couple of days in 2014. And when I first started my doctoral studies, we threw around a lot of different ideas. Some of you know I thought about studying hornbills, and then I realized how long it takes them to actually reproduce. And they also nest in trees, which didn't turn me away from woodpeckers, but whatever. Hornbills are hard to study, and I didn't end up doing that. But at some point, he and I were just talking about woodpeckers. Man, it would be cool to study one of these tropical species that we don't know anything about, where there's some indication in the literature that they might be cooperative breeders. Well, this species isn't a cooperative breeder, but I do think it's super interesting. And if it weren't for Walt and all of his insight and patience with me and all of the feedback he's given me over the years and the encouragement, I really wouldn't be the scientist I am today. So hopefully, Walt, you can still hear me. Thank you. And now I will quickly dedicate my dissertation to my mom, who is here today. She came all the way down to the terrible field site I was working at in Cozumel in 2008 and visited me while I was studying black catbirds. To today, where she's a proud grandparent of our beautiful son. Thank you, Mom, for always encouraging me. And my dad, who also came down to Cozumel that year. You can see where I get my sense of humor from. Both of my parents have always been super supportive, though neither of them are biologists. I used to bring home animals all the time. I think they knew before I did that I was going to be a biologist. So thank you. Next, this guy, who is in Ithaca today. This is my son Michele, who was born on August 3rd, 2017, so right at the auspicious time of when I was trying to finish. Try finishing a dissertation with a kid. It is a challenge. But he is awesome. He has my sense of humor and Conchetta's sense of humor, and he's super awesome, and he likes birds. Lastly, almost lastly, my wife, Conchetta. Been through a lot. Conchetta and I actually met right before I started at Cornell. So this is a picture of her helping me move in. I think we had gone to the beach like that weekend, so we're nice and red and sunburned. So she's been with me from the very beginning, where I was a very green and much hairier scientist, at least up here, to some of the worst and also best moments where we thought there wasn't going to be much more of us. And so you've been everything to me, and I couldn't have done this without you, and I'm trying really hard to make you cry, but not trying to. So thank you, and I love you. And lastly, I want to thank PBO 516. This tree. So much to say about this tree. Quick background on this tree. This tree died in between 2015 and 2016. I mentioned before that when trees die, they tend to become colonies. This is one of those data points, and it was awesome. It was like no cavities on the tree. It just died. Let's see what's happening. So I climbed this tree in 2016, and for those of you who don't know, I got up to about 45 feet. I was finishing up, and oh man, things went bad real fast. I'll spare you all the details, but I got medevac to the US. I still have some metal in my body, and there was a sepsis scare. It got bad, but I'm really happy to be here today and to say that in 2017, I went back and I climbed that tree so many times, much to the embarrassment of my family and friends, and I'm sure the worries of my wife, but I got so much data from that tree that year, so I think I didn't die, so I guess it was worth it. And with that, I will take any questions with the time that remains. Thank you so much.
Couple minutes for questions.
Yes.
All woodpeckers solitary feeders.
Solitary feeders? I think there's some indication that Indian flickers forage socially, but hardly anything's been published about it. I know in acorn woodpeckers they have the granaries. I'm not sure about social foraging in other species. I don't think that this species does it. Outside the breeding season, sometimes you'll see them aggregated on fruiting trees, but I don't have the data yet to know if those are actually like colony members that have stuck together and are following each other. Certainly when you sit and watch a colony tree, the birds are going off in all different directions. And they're super omnivorous. They feed on local fruits, insects, sometimes vertebrate prey as well. So I think it's unlikely that social foraging would actually be a benefit to them. It's a great question. Andre?
So you showed that live and dead trees have different mortality causes. Flooding or not flooding?
For the nests.
The nest, yes. And you showed that, you said actually that there's enormous variation between years as regards how much rainfall there is.
Yes.
So have you considered analyzing the data for nesting mortality or nest loss in dry and in wet years?
I have. I wasn't sure if I had enough data to do that. And if you have suggestions for what would be the best way to tackle it, I'd be very interested.
Because it could be that the selection pressure varies between years as regards what kind of nest to use. That's why you don't find this kind of relationship as you expect.
Yeah, that's a really good point. Brian?
Are these guys just bad natal dispersers? Do you find that relatives live next to each other, or there's film patry on trees? You know?
Yeah, you had to ask that question, huh? So the short answer is I don't think so, but we've banded over 300 nestlings, I think, over the years, and only 6 of them have come back as breeders. So I don't know if that means that a lot of them are dispersing or a lot of them are dying. We really confine our activities to the study site. So I do have the SNP data, and so I should do the relatedness analysis to understand that. And that is high on my priority list for the next thing to do. Chris?
Are cavities ever the cause of mortality of the trees? Is there pressure to continue to build in live trees despite the effort because it will kill the tree faster?
That's a really good question. If you were to ask the Dominicans, they would tell you that the woodpeckers are killing trees. But the answer that I would give you is I don't know. It's entirely possible that they cue in on like subtle defections and like maybe fungal infections that we just aren't seeing. But yeah, I'm not sure. I think it would take a slightly more nuanced approach and more intensive work on the palms themselves to understand that. But it's a really good point. Yes.
You said that there were many cavities on one tree, right?
Yes.
So when a woodpecker leaves and wants to come back, how does it know where its cavity is from all the other cavities? Do they sometimes go together by mistake if there's an aggression?
So my funny answer is that we mark all the cavities, and so that's how they know how to do it. But the real answer is most like almost all of the marked birds that we've watched go to their nests, especially when it's an active nest, not just, when it's empty before they're about to lay. But we do see intruder birds. So birds that aren't the social parents that come in. Sometimes they'll even go into the nest when there are nestlings. I'll try to answer your question because I'm going to go off on a tangent. I don't know exactly what cues they're using. The colonies aren't that large, so it's probably a relatively easy task for them to keep track of where they're putting themselves.
But you never saw like a red-banded one go up, but it went down. You never saw some event like that.
I've rarely seen that. Usually that's when there's an unmarked bird coming onto the colony tree, and it's probably just too close to the nest, and so it like goes up and gets chased off.
Maybe time for one more question.
Rusty?
How long has the island been around, and how long have the woodpeckers been on the island, and how's the habitat? Is it, for example, been pretty stable over that time period?
Man, those are super great questions. Can't remember the age of Hispaniola, but millions of years at minimum, because there are like antler specimens like going back a long time. People have been on Hispaniola for over 1,000 years. Europeans have settled it since Columbus arrived. So I imagine that the land use patterns have changed a lot, but royal palms are native, Kana palms are native or endemic. And so I think there probably has been some impact of human activities that have shifted maybe the prevalence of coloniality. But yeah, if you read some of the earlier research that people have done on the species, they suggested that because the species is so size dimorphic, they lost territoriality because there was no benefit to being territorial, and so birds were just like, I don't have to defend anything, I'll just nest near other species. Does that answer your question?
Yeah. The question is, these guys are weird among woodpeckers.
Yeah.
Are you trying to infer some relationship with the habitat at present day, when in fact it might actually be different? The factors.
Yeah.
You got at it.
I mean, I think the unfortunate thing is because coloniality is so rare in the woodpecker family. I mean, it's evolved once in Melanorbes, once in Calaphes, maybe twice in Melanorbes, if you look at Lewis's woodpecker. There probably just isn't enough variation to do the comparative analysis that would really answer that question. Yeah, it's a great question. Thanks.
Okay, great with that. Thanks.
Thanks for coming. Yes, they will, but you'll also see them using cavities that are promoted. So I don't know how the birds that have a nest are occasionally.
There are only a few colonial areas.
Yes.
And most of their invading the U.S. They're on the islands, and they build this massive nest of six. And each pair has its own little cavity. And to my knowledge, nobody's so you end up getting a job or whatever. So
I do have a postdoc.
yeah, where monks are.
Yeah, thanks. Okay. Hey, buddy. Congratulations. Good to see you.
I kind of went on autopilot. I don't remember most of what I said.
It's just, hey, you're doing great now. Thanks. It's good to see you. I'm good, man. Good. Good luck. This next part is fun.
Thanks.
And
I'm kind of postdoc, so.
Clean
This is the big one.
I mean, stand there and let her explore. You know, and then he's been, she said, most people like, oh, right. It's not even.
Yeah, we're going to.
Yes, yeah. We're just gonna be there in like 10 minutes. Time to- Supposed to bring treats for you guys, or we just get that after? You have to mail them.
No, we're good.
We'll make it feel better that you guys didn't get it.
Yeah, exactly. I want them to sum up.
Yeah. Eyes on you.
So I'll see you.
Yeah.
Exactly.
So, my building.
Oh,
So,
really?
isn't that, so that's.
Yeah, he doesn't like
Fair enough. Is it a family base? I don't want to interrupt your
I just.
Yeah, definitely.
Like, this is
Her
just.
husband's name is Chad.
Yeah.
Her family's also from Sicily.
He's not just.
I'm not Sicilian.
Oh, really?
Yeah, that's,
But you don't
my
know
sister.
Sicilian.
Oh, my first name is the other grandmother's name.
Because it was.
As a I was very Yeah, I'm 4 years old.
Yeah.
It's like.
You know, it's thanks to me that you're not considered a Carmella, because Catholic families did that back then.
So her maiden name is Miguel. So my last name is LaPergola, so now it just sounds even more. And our son, we wanted to stick with an Italian name. Michele Giuseppe LaPergola.
Oh, it's wonderful. So you're right here
It's good fit.
It's been a long time. Yeah. Well, my uncle dug into it at one point. He thinks that it was originally LaPergola.